You are watching a demonstration of Biopod, an exciting new software solution recently released by BioNeos Incorporated. Biopod is designed to speed the mutation discovery process by harnessing the knowledge of many resources into one concise location. Use these resources combined with a unique search algorithm to help you find mutations faster, and provide the necessary tools you need to perform your research faster and more efficiently. Before showing you the application, let's first explore the unique search algorithm that sets Biopod apart from any software you've seen. When searching for novel mutations, investigators typically use what we call the serial approach to designing PCR-based experiments. From a locus interval or a set of genes, a ranking is given to each gene. Then, progressing from the first gene to the last, experiments are designed to cover the entire gene serially 5' to 3', spanning the complete coding segment of the gene. This process continues until the mutation has been found, or, even more often, until all resources have been used. The time and cost for each experiment quickly adds up with a large tissue set and list of candidate genes. A new, smarter technique may be used which utilizes a vast amount of data collected from previously discovered mutations. This technique is called the progressive prioritized approach. Quite different from the serial technique, the progressive prioritized approach designs experiments at the best possible location of every gene in the candidate list. If the mutation is not found, the second best location of every gene is examined, and so on. This allows for much broader coverage, yet uses less costly resources. This begs two very important questions. How do you choose the best location to screen? And how do you manage where you have already screened and where you have not? Biopod is the answer to both of those questions. When you open Biopod, you will be presented with an empty window that is ready to load a candidate gene. Simply choose Load from the candidate menu. Here we will search for the gene. You may choose to search by locus, or search by a keyword such as part of a description, an identifier such as a RefSeq ID, or a gene name. Here we will assume that CD34 is the first ranked gene in our candidate gene list. My search for CD34 has returned one result, and I can verify it is a gene I want by looking at its chromosomal location, number of transcripts, size, and so on. It is also important to note here that although I am searching a single data source, Ensemble in this case, Biopod is very flexible and can interact with many online data sources and even your own local proprietary data source given the proper plugin. This makes Biopod a very powerful and versatile tool for any research laboratory. Once I click Open, Biopod will contact all of my specified data sources to gather information about the CD34 gene. For example, right now it is getting gene structure, domains, and SNPs from Ensemble, predicted secondary structures from NNPredict, and transmembrane domains from Phobius. This would take a long time by hand, but Biopod does all the work of gathering, sorting, and aligning the data for you. Now that the gene is loaded, you will notice two primary windows. The top window, called the transcript view, shows the mature RNA with the introns spliced out and all coding-related annotations such as domains and secondary structures. The bottom window, called the genomic view, displays the entire gene including introns and up and downstream sequence. It shows all annotation that is not specific to the coding portion of the gene. Each type of annotation is on its own annotation track. Mouse over the track to see what kind of annotation it is. If the track has overlapping annotation, you may compress the track into a single layer or hide it altogether. You will also notice two graphs, one for the mature RNA and one for the entire gene. This is the output of our unique search algorithm called PAR, which implicates regions of high interest when searching for disease-causing mutations. A higher point in the graph suggests that region to be more important in your mutation search. The highest point is conveniently highlighted by a green bar. You could verbally translate this algorithm as saying, start your search for mutations in this exon. Likewise, you would also start at the green PAR region of your other candidate genes. If you want to see two PAR regions for a gene, simply make the change to the PAR options. Now that you're familiar with loading a gene and the general layout of Biopod, let's take a look at other key features that make Biopod and the search algorithm easy to use. Biopod makes a gene very easy to navigate. If you want to take a closer look on a nucleotide by nucleotide level, simply zoom in. Here you will see the nucleotides as well as the amino acids within the coding region of the gene. Also notice the informational panel that tells you the numbered position of the amino acid and nucleotide within the gene, the genomic location, and the PAR score. Now let's move to one of the PAR regions of this gene. A portion of sequence can easily be selected with the mouse or use the pop-up menu to select an entire exon, an entire coding region, or the entire gene. Normally, we would select a smaller portion of sequence such as the PAR region, but for the purpose of this demonstration, we will choose to select the entire gene. Once you have a region of sequence selected, you may perform many different tasks on that region. For example, you may copy the sequence, either nucleotide or peptide. Even create your own custom annotation. One of the nicest features is the ease with which you can span the region with PCR primer pairs. This primer picking functionality is a graphical interface to the PrimerViewer program developed by the Whitehead Institute for Biomedical Research. Choose to leave the PrimerViewer parameters at the default values or change any of them. For example, we will change the minimum product size to 150 nucleotides and the maximum melting temperature to 60 degrees Celsius. You may also choose to avoid picking primers on a known SNP. Once you are satisfied with your selection, click the button and the primers will instantly be generated. Whether you are picking primers to span an entire gene as we have done, or a small portion of selected sequence such as the green PAR region, the primers are generated very quickly. Zoom in to see the primers more closely, or right-click them to get more details. You may also mark them as succeeded or failed to be remembered for future experiments. If you added a data source to Biopod that interacts with your own local data system, you may save the primers to that data source. Other options include an export wizard from the primers menu, or save everything to a file from the candidate menu, which can then be opened later. Biopod makes a new progressive prioritized screening approach possible by answering the two important questions mentioned earlier. The question of where to screen is answered by the green PAR regions powered by our unique search algorithm. The question of management is answered by the ease with which you can design primers, save them, and mark their success status for future reference. For more information on the PAR algorithm, you may be interested in reading this article from Human Mutation titled Prioritizing Regions of Candidate Genes for Efficient Mutation Screening. It was published in Volume 27, Issue 2. If you have any questions about Biopod, please feel free to contact BioNeos. Visit bioneos.com for contact information and other details about the Biopod software. Thank you again for your interest in our rapid mutation discovery solutions.